Can you hear me? Hey, I don't like using these microphones, but I was told I have to. Welcome, everybody, to our annual Riser Lecture. I see lots of familiar faces. This is our biggest academic event of the year. We're very excited to have you all here. We're particularly excited to have our speaker, who will be introduced in a moment. As you all know, we've been doing this for a while, and I always like to give you guys an update on where the Marine Science Center is. And I think a lot of you who've been coming here for a long time, people like Peter Focal, the audience is a lot bigger now, isn't it, Peter? Yeah. So, obviously we have a much bigger community here, and as a result of that, the science that we're doing is having a much bigger impact in the world. And as I may have told you about last year, over the past couple of years, this urban coastal sustainability initiative that we've been working on, which is really used to try and hybridize interdisciplinary solutions to create cleaner, safer, smarter communities. I feel like a lawyer now trying to doing a commercial, you know. But that's our tagline. And we've had such success that the university is now turning this initiative into an institute, which is great news, right? You can give a round of applause. And of course, as part of that institute, we're always trying to find new talent to bring here to help us with this. And is Stephen Cyphers here? Stephen, where are you? Raise your hand. Is he here, John? Well, we've hired two faculty. Guess what? Guess what? He's an assistant professor too. He's screwed. So this past year, we've hired two new scientists, and the great thing about them is they're outstanding scientists, but they're even better people. One of them is Stephen Cyphers, who works in the space of socio-ecological coupling. He's an assistant professor, and he was an NSF postdoc here before we decided to make him official. And then the other person is Dr. Jennifer Bowen, who will be introducing our Riser lecture in a moment. And I'm very excited about having Jen Bowen here because she's an associate professor with tenure. She came here from the University of Massachusetts at Boston. And Jen is one of these folks that does great science, but has also quickly assumed a real leadership role in our department. And she's been really key in helping us with strategic planning and also developing our new major in ecology and evolutionary biology. So these are the kind of folks that we bring here. Let's give a round of applause for Steve and Jen. And thank you. And so Jen's going to come up and introduce our speaker, who I'm very excited about is here, but Jen's going to say all the embarrassing words and show the embarrassing pictures.
Thanks, Jeff, and thank you all for coming today. It's great to see such an amazing crowd, and I'm really thrilled to be a part of the Northeastern community. And I'm also thrilled today to be able to introduce today's Riser lecture speaker, Dr. Linda Deegan. I've known Linda for many years, and she has been formative in creating the leader that I am today. So Dr. Deegan actually completed her undergraduate degree from Northeastern University a few years ago. Her honors thesis was titled The Growth, Production, and Metabolism of the Mummy Chug in a New England Salt Marsh. And this is work that she did under the direction of Dr. Ruber, who was a good friend of Doc Riser's, after whom this seminar series has been named. And I don't think that Linda fully realized when she was doing her honors thesis that some of her most seminal work would be done in these very same salt marshes that she studied as an undergraduate. So if there are any undergraduates in the audience, pay attention to your honors thesis now, because you may have a lifetime career of that. So this is Linda in one of the marsh ponds at Plum Island, probably looking for the very fish that she studied as an undergraduate here many years ago. After she left Northeastern, Linda went on to complete a master's degree at the University of New Hampshire, and she completed her PhD from Louisiana State University, working with John Day. After completing her PhD, she spent a couple years at UMass Amherst as a professor before moving to the Ecosystem Center at the Marine Biological Laboratory. And she's now a senior scientist at the Woods Hole Research Center, and she has had an incredibly accomplished career. She currently has, and I had to triple check this because it seems incredulous to me, 6 actively funded National Science Foundation grants, totaling almost $10 million worth of revenue across all collaborators. She's published over 120 manuscripts, including several in our top journals, including Nature, Ecology, and the like. She's received countless awards, including a prestigious Fulbright Award to do research in Brazil. One thing about Linda is that she has research programs that you will hear nothing about, one in Brazil, one in the Arctic. So she's extremely diversified as well. She served on numerous expert panels. She was an NSF program officer. She has mentored 10 postdoctoral fellows, 16 graduate students, countless undergraduate researchers, and has just had a tremendous influence on the next generation of ecologists, including myself. So as I mentioned, I have known Linda since before before I started working for her as a postdoc, and on this project that she's going to be talking to you about today. So this project began in 2004, which is when I started working for her, and it's a remarkable testament to Linda's mentorship that over the 13 years of this project, there have been 5 postdocs working on this project, all of whom are in tenured or tenure track positions at large research universities like Northeastern. Linda's going to tell you about the project in detail, but in advance of that, I want to share some insight into Linda's personality that can be gleaned from this particular photograph. And this is what the postdocs and technicians who have worked for Linda refer to as her PI pose. Although usually it comes with her hands on her hips. And when she gets this pose, and while she's in the marsh, we know that all of our plans are about to explode. And that's both good and bad. Good because it usually means that she has come to some insight that we had not previously considered about our experiment and is going to change the way that we think about marsh ecology. Bad because we know it means that we're going to be in the field a few more hours than we thought that we were going to be. But it doesn't matter because in the end, when we get home at the end of the night, Linda's there to cook a giant spaghetti supper for everybody on the team. And then she goes and does the rest of her work for several hours while we go to sleep and get ready for the next day. So she's been an extremely impressive role model and a good friend, and it's my distinct honor to introduce Linda Deegan.
Sure who that person is that Jen just introduced, but thank you very much. You know, I really am honored and delighted to be here today. I think there's at least 1 or 2 people in the audience who understand how much Nahant has changed since I came here as an undergraduate. And I'm really thrilled to be here and thrilled to see all the changes and the successes that the Marine Station has had over the years. Dr. Reiser was really a well-known marine ecologist when I was an undergraduate and worked with Dr. Ruber and influenced a lot of the people at Northeastern who influenced me. So it's exciting to be here. I'm going to talk to you today, as Jen said, about some of the work I do in coastal marine ecosystems, particularly in salt marshes. But I want to tell you just a little bit about myself so you understand why it is I do what I do and the approaches that I have. And I will say that, you know, when I was thinking about this lecture, I told Jen I was really sort of intimidated. I imagined coming to a room full of strangers and trying to explain this complex experiment, of which most of the data isn't mine, actually. It belongs to other people. And I thought I'd be looking at a sea of unfamiliar faces. And it turns out, after having spent here for this entire day, meeting all the graduate students and the faculty, and I feel like I'm right at home with people who do the kind of work that I do and do the kind of work that I think is really important. And, you know, if you want to put it in science speak, these are the kinds of questions that I try and think about. How do organisms interact and affect ecosystem function? How does the way the landscape is structured and the environments they live in affect their ability to live and thrive and reproduce? And what are those feedbacks between the animals and the system itself? And more, I've really come to think that understanding and being able to predict how the environment and the organism is going to change in response to large-scale drivers is a moral obligation of us as scientists. And in particular right now, I think climate change is one of the biggest drivers that we have to really face and provide good science so we can, as a society, make good decisions. So that's what I do in science speak. But how did I get to this point? Well, honestly, this picture right here encapsulates it all. This is me, 10 years old, living on an estuary in Alabama with a mess of speckled trout. At that point in my life, I was hooked, as we say. And my parents probably could have told you at that point that I would turn out to be an aquatic ecologist and I would study fish. Some would say I obsessively study fish. Because for me, it's all about, you know, taking a look at aquatic ecosystems, lakes and streams and estuaries, and thinking about how those systems work from the eyes of organisms that need everything underneath them in order to survive and thrive. And that's what I try and do in my work. Now, at first, I was just interested in the fish. So I did an undergraduate thesis with Dr. Ruber. and it was all about the mummy chog in the marshes. But really, as I continued on in my studies, I began to really realize you had to know a whole lot more about the environment this organism lived in and the other organisms that it lived with in order to be able to understand the fish. And so over time, I really sort of evolved from what you might think of as a classic fish biologist, ecologist, into an ecosystem ecologist. And now I work with a lot of different people in a lot of different ecosystems. People who look at geomorphology, who look at genomics, who look at energy transfer, primary productivity, all really, it's still for me all about the fish, trying to put together that ecosystem that creates the environment in which organisms can thrive. Now, why salt marshes? You know, as Jen said, I've been privileged in my work to be able to travel across the globe, working from the Alaskan Arctic down to the Brazilian Amazon. Again, it's all about the fish. But really, I'm always drawn back to the salt marshes of coastal New England. And this is an area, this is an ecosystem that I was introduced to by Dr. Ruber when I was an undergraduate at Northeastern. I transferred into Northeastern, and as a junior, was searching for somebody to please sponsor me for an honors thesis, because it was really important, and that's what I wanted to do. And I knew I'd choke up I knew I'd choke up if he showed up in the audience. You know, he was incredibly gracious and showed me Plum Island and showed me mummy chogs and really helped me do a project that I was really proud of. You know, mummy chogs are this tiny little iconic minnow. This is about how big they are. They're everywhere in salt marshes by the thousands, from Canada down to Florida. But as I think you'll see in this presentation, they really, for a small animal, play an outsized role in coastal ecology and in our understanding of coastal ecosystems. And Jen's absolutely right. I always warn my undergraduates, be careful what you choose for an REU project or an undergraduate thesis project, because it can haunt you for years. Fortunately, I really like working on mummy chogs and salt marshes, so I'm okay with it, but keep that in mind. So the reasons that I love salt marshes are really a lot of the same reasons that most people love salt marshes, and most of you know all these reasons, right? They're very, very productive. We find them, if you're a naturalist, to be sort of beautiful, peaceful places to go enjoy the natural world. And they are one of the world's most productive ecosystems, and this provide lots of habitats for sort of iconic organisms. Here's my little mummy chog here in the mouth of a snowy egret. So participating in the food chain, this very productive food chain, which is one of the things that they're very well known for. The other thing that salt marshes are known as is hotspots for fishing. And this is my long-term colleague and co-principal investigator of the tide experiment, Bruce Peterson, with my niece Erin, who we're trying to teach to really, you know, appreciate the bounty of estuarine ecosystems and pass on to the next generation that dream of a system that's bountiful and plentiful and still provides a lot of those natural resources to the world. Now, salt marshes, because they sit at that interface between land and the ocean, also do some ecosystem services that you might not think of right away, or at least we certainly didn't think of them 30 years ago when we were thinking about salt marshes. One of them is that they provide a buffer to our upland development here from storms, and sometimes these storminess by attenuating wave heights. Protecting upland systems from coastal storms is one of the things that salt marshes do. This is the kind of thing like food web production and fisheries, storm protection, that we call ecosystem services. The other thing that salt marshes do that's really critical is they protect the coastal ecosystems on the other side of them. So the eelgrass and the mudflats from activities in the upland. And they do that often by acting as what we think of as nutrient filters, right? So they take the nutrients in runoff and they convert it into plant biomass, and that helps protect other coastal ecosystems that might be more sensitive to nutrients, like eelgrass, from the effects of nutrient loading. And this has been a very powerful motivation for why we want to understand and preserve salt marshes. But quite honestly, the world has changed a lot. When Dr. Ruber first introduced me to salt marshes, there were 4 billion people in the world. Now there are 7. 7 billion people in the world, and most of them are living, most of them are living in coastal regions, right? This is a very classic photograph from space of lights on Earth, and you can see that almost half of the world's population lives within 60 kilometers of a coastal region. That's a lot of people in a very small area, and that small area is of incredible importance for all of these ecosystem services that we talked about. Back in the bad old days, the things we worried about were a lot of direct impacts. There was a lot of dredging and filling of salt marshes for human development, right? Back Bay, Boston, classic example, glorious salt marsh no longer. A few very nice colleges sit on top of it, but not so much the mummy chog, right? Mostly, for the most part, I think we've taken care of those kinds of impacts because we now realize the importance of these areas. And salt marshes are facing, instead, a lot of sort of slow, more diffuse, more non-point impacts that are affecting them, like higher temperatures and rapid sea level rise and stormier, harder, more intense events. And all of those are affecting and stressing coastal ecosystems. But really, the biggest impact that humans have had in coastal ecosystems is nutrient loading from upland development. So this is a map here that I developed with my colleague Will Wollheim from the University of New Hampshire and Bruce Peterson and several other people, looking at nutrient loading to the coast since the turn of the last century, so since the Industrial Revolution here. And one of the things you can see is that most of the nutrient loading indeed is associated with high population centers. Most of that's in the temperate zone where salt marshes are located. So all those little dots are places in the world where there are large salt marshes and there's lots of nutrient loading in those areas. And it used to be that we thought about the nutrient loading going to the coast as deriving from agricultural. So farmers were all to blame, right? And that's still true in a lot of places. There is a lot of agricultural runoff. But in a lot of places in the world, the source has changed from agriculture to human development, from sewers and septics that support the megacities along the coast and sprawling suburbia. And this high nutrient loading has resulted in the classic eutrophication syndrome, where you've got high nutrients causing algal blooms of one sort or another, decaying organic matter, low oxygen, fish kills. Those are a very classic effect in coastal ecosystems of eutrophication. And so, again, we go back to this idea that because marshes occupied these large areas along coastal systems, because they were very high productivity and they took up nutrients, responded to that nutrient to increase plant production, so taking up nutrients by plants that's then used in food webs, making all those fish. And there are also sites of microbial processing, particularly having very high rates of a microbial process by which microbes use the nitrate, turn it into basically an inert gas that goes off into the atmosphere. We really thought that for a long time that a little nutrient loading to salt marshes might actually be kind of helpful, because as we increased the plant production, it actually stored carbon in the peat and allowed them to better keep up with sea level rise. Here we are stimulating a food web, meaning more fish. And all of that was maybe a good thing as long as we didn't push it too far and drive it anoxic. And really, whether or not we drove the marshes anoxic was what we considered to be the key issue there. And based on a lot of individual processes, or a lot of individual studies on small plots and all of these different processes, we really thought that marshes might be sort of the solution to our eutrophication problems in the coast. And so this was all very logical, put together by multiple pieces of evidence from multiple experiments done by multiple people. But we really wanted to ask the question, is this sound okay? Because it sounds like it's going in and out and in and out. It's okay? Okay, I'll stop worrying about it. We really wanted to ask the question, were we right? If we put together all these different pieces of information taken from different sources and different perspectives, could we predict how an ecosystem would actually respond to this change? And what were the limits to what a salt marsh could do for nutrient retention? And so I gathered together a bunch of my colleagues from a lot of different disciplines, and we wanted to test this idea. So we actually went then up to Plum Island, and we chose Plum Island for a site where we're going to add a lot of nutrients. We chose it for a very good reason, which is it's one of the very few not very nutrient loaded estuaries left in New England. So it wasn't very nutrient loaded, so we could do a manipulation against a background that was sort of typical of almost pre-industrial conditions in salt marshes. And we designed an experiment, and as part of this experiment, we wanted to look at lots of different components of the ecosystems, and we wanted to look at the interactions of all the different habitats within an ecosystem, within a salt marsh. So salt marshes have multiple habitat types, right? They've got marsh creeks, they've got streamside Spartina alterniflora, the stuff that goes really tall next to the edge, and then up on the back marsh here, we have the high marsh. And we wanted to understand how that streamside plants might interact with the back marsh, that high platform marsh. And of course, for me, I wanted to understand the fish. So we wanted to study everything from the tiniest of microbes that Jen works on, all the way up to that big monster fish, the mummichog. And so we chose experimental sites that were basically the size of the home range of mummichogs. And we did some early studies, and we showed that in these systems, a creek length that was about 300 meters long, encompassed by tagging mummichogs, we showed that was basically their creek summer home range. So we found primary creeks in Plum Island that were about 300 meters long, and then looked at how much of the marsh that actually flooded, and the fish moved out onto to accumulate energy and come back down into the creek, because the mummichogs move from in the creeks up onto the marsh to occupy and use that habitat at high tide. So we needed this whole ecosystem to support those fish. We also decided a couple of other things. So we needed these large areas, and those large areas turned out to be about 60,000 meters squared, which actually turns out to be 10 football fields altogether for one experimental treatment. So that's a really large experimental treatment, but it's the size we needed to capture the complexity, the habitats, and the home range of the species that we thought were really important. So we're there with 60,000 square meter units. And we also then thought about a couple of other things. One of the things that we thought was really important was to add the nutrients the way that eutrophication would affect a marsh, which meant we wanted to add the nutrients in the flooding water, because we knew the nutrients are coming in the flooding water, and they're going to interact with different parts of that ecosystems at different rates, depending on how often they flooded, right? So the creek flooded absolutely every tide. The creekside Spartina flooded pretty much every tide. And those high marsh areas are only flooded about 10% of the time. So we're going to have a gradient of nutrient loading there across that platform. And we thought that was really important. We also thought it was really important that the nutrients be dissolved in water, because that's how it gets to the marsh in reality. But we felt that nutrients dissolved in water would interact differently with the ecosystem than if we just went out there and sprinkled dry fertilizer across the marsh surface. And that is how really all of the prior experiments had been done. The other thing that we thought was really important was to add nitrate and nitrate only. Now, nitrate is the major form of nitrogen that moves from uplands into the coast, and is what stimulates nitrogen, is what stimulates all the productivity. But we also knew that ammonium and nitrate interact differently with different, particularly microbial processes, but also plant processes. And most of the other experiments that had done a combination of ammonium nitrate fertilizer. They did that because if you're lugging 50-pound bags of fertilizer out in the marsh, you get a lot more nitrogen in an ammonium nitrate bag of fertilizer than you do in a calcium nitrate bag of fertilizer. You get almost twice as much. So you carry half as much. But we thought it was important because of the way those two ammonium nitrate forms interacted with the ecosystem. I have to say that this turned out to be a clever decision for another reason, and that was shortly after we started this experiment, Homeland Security started registering all the people that were ordering ammonium nitrate fertilizer. So those were some of our rationales and why we designed the experiment the way that we did. And as I said, we wrote this proposal, and this was my first real attempt at leading a multidisciplinary, interdisciplinary, large. We had 8 principal investigators on the first proposal, and they were all really strong people and brilliant. And I was convinced, I tell you, that we were going to be funded on the first try. And that's a dead giveaway. We weren't. It was brilliant, I thought, but I was crushed when the reviews came back. So for all of those postdocs out there who've been crushed by your first reviews, don't worry about it. Life goes on. We spent actually the next 3 years rethinking, rewriting, and re-explaining what we wanted to do, because the biggest comment that we got from reviewers was, what do we need to do another marsh fertilization experiment for? Don't we already know everything we need to know? And we had done a pretty good job of explaining everything we knew up to that point, and we hadn't done a very good job of explaining why we didn't think that what everyone had done in the past was sufficient to answer the question. Yes, we'd learned a lot, but we didn't know everything we needed to know. So it took us 3 tries before this experiment finally got funded, and we got the reviews that we finally needed. I distinctly remember that one of the reviews said, these people are crazy, but if anybody can pull it off, they can and they should. And so we were funded for actually a 5-year proposal the first time, which was very useful because a lot of what we were studying took a long time to sort of develop and really understand. So as I said, we cheered when we got funded. So this is one of the groups. This is actually one of the later groups. And just to give you a sense of how the dimensions of this experiment, we targeted 70 micromoles nitrate in the flooding water. And we chose that level because that's a level of nitrogen which was known to induce algal blooms, oxygen kills, and oxygen deficit and fish kills in eelgrass and open bay areas. So it's designated by EPA as a moderate to high loading level, right? This isn't even crashing through the top of what we've done to a lot of our coastal ecosystems, but it's up there, right? We wanted to make sure we got some response. And to translate that, which was shocking to me, that loading rate meant we were actually building the equivalent of 1,000 houses in the watershed of a marsh area, which, compared to urban Boston, is not that many houses in the watershed of each of our experimental units. This took, it turns out that it took a trailer truck, 2 18-wheeler deliveries of nitrate stacked high on a trailer truck every year to deliver 40 tons of nitrate. 40 tons of nitrate is what we needed every year to pull off this experiment, which, if you're not impressed by that, you should be impressed by the fact that we had an experimental manipulation that cost $40,000 a year. And NSF agreed this was important to do things at this scale and this big. And then, there's sort of 2 humorous components to this experiment. It was large, it was audacious. And it turns out people would drive, so I work up at Plum Island at a field station that's converted from an old farm and farm stand. And people would see the nitrate delivered, and they'd come knock on the door, and they'd say, when's the corn going to be ready? And we'd have to say, no, this is a science experiment. And they're like, what? And the other thing is, I was reminded at lunch today, we actually ended up competing on the international market with farmers to buy this much fertilizer. And it turns out that most of the fertilizer, nitrate fertilizer, is prilled from Chile. And there's like one salt flat where they mine it and they turn it into bags of fertilizer, and then they put it onto boats and they ship it up to whoever wants nitrate anywhere in the world. And when Chile had the earthquake about 5 years ago, our nitrate delivery stayed in Chile because it didn't come anywhere. And I spent many a night on eBay bidding against farmers in Iowa for nitrate. Not an experience anyone needs, but there we go. Wait a minute, where am I? Oh, yes. So our plan was really pretty simple. We wanted to manipulate, measure, and model, right? And that's something I heard all day long today as I talked to students. Modeling is a key component. A lot of the students are doing experimental manipulations and tests of hypotheses, and that was really critical. And the other thing is that this experiment was very complex, and we needed a multitude of different expertise in order to do it. So we brought in as colleagues hydrodynamicists, people who studied sediment transport, people who did biogeochemistry, plant community and population ecologists. Jen and John Hobby joined us to look at microbial processes. We looked at benthic algae. We had population and community ecologists, from people who studied small invertebrates up to people who studied the gigantic mummy chog, the top of the food web there. And actually, we even branched out into birds one year, which was kind of interesting. And we used a variety of, embedded within this larger scale experiment, we used a variety of techniques, including large scale stable isotopes to track the added nitrogen through the food web and through the biogeochemical processes, and lots of small scale manipulations. Who was it? There was a student telling me about the crab fights that somebody was doing, looking at competition. So we did a lot of caging experiments to answer population biology questions as well. And every year in May, we'd go out to this beautiful section of marsh and basically muck it up by dumping in 40 tons of fertilizer. We toasted every year that first batch of 40 tons of fertilizer, which we had to haul out 40 bags every 3 days, 50-pound bags every 3 days out to the marsh, mix it in this 1,500-gallon tank, and then we had a computer-driven program that pumped it into the marsh creek in proportional to the hydrodynamically determined cross-sectional flux of water to try and achieve, as constant as we could, a nutrient level at around 70 micromolars. And we toasted it with mimosas on the marsh every year. And this is actually the 2016 field crew that was out there in March with the mimosas on the marsh. And then we also went back into the lab, and we kept doing this for 13 years. And every year we developed this really detailed planning schedule of what we were doing every day of the week, when you were sampling what. Mostly we did this so that we'd know what we should have been doing when plans didn't go exactly the way they should, or the PI walks out into the field and goes, let's do something different today. There was 1 month I was actually banned from coming up for that reason, but that's another story. By the end of the summer, we were usually sunburnt, greenhead bitten, and much, much stronger. And we have done this every year for the last 13 years, including the year of the Chilean earthquake. And really, we learned a lot of things from this experiment, a lot of unexpected things. Some things were sort of expected. We learned, for instance, that when you work in marshes, you get very, very muddy. So this is David Johnson, who was one of the first graduate students on the program, who has graduated now to being a postdoc, and then he was a PI. This is Carrie Galvin, one of the postdocs who's our graduate students, who is now a faculty at a university in Chicago, in Illinois. But you get muddy, and Ashley reminded me today that we were simply ahead of our time. For those of you that haven't seen it, Nordstrom is now selling pre-muddied jeans for $400 a pair. So I think if we just take all the jeans that we've muddied up, Ashley, we could probably pay for your dissertation. We also really learned a couple of other things. One thing is sort of trivial, is never wear knee-high boots in a marsh because they won't come back with you. But the more critical thing we learned was the importance of having good colleagues who will both pull you out of the marsh when you get physically stuck before the tide comes up, and also pull you out of those quagmires when the science and the data doesn't really quite do what you think it's going to. So you've got people to talk to and people to work with who would approach things from a different point of view. So that speaks, I think, to the power of teams and thinking about things together. And over the course of the TIDE experiment the last 13 years, we've had well over 100 scientists come and work there. We put together this experiment as what we felt was a platform for the open community, and lots of people came in. And these are just some of my key colleagues, and I'm going to tell you some of the results from this. I can't do justice to all of this, not even close. But these people are some of the key colleagues who've worked with us most recently, and some of them you know. Jen Bowen is now here at Northeastern. So what I want to do is just run you through some of the key characteristics from about 4 different components of the system. And I'm going to go through these pretty quickly. And as I said, you know, a lot of this data is not mine because I didn't necessarily take the data. It's my colleagues, and I'll try and acknowledge them as I go along. But really, my job was to herd the cats and try and keep us all going in the same direction and not trip over each other when we were out there in the marsh. And I worried about the fish. So one of the first things we wanted to do was test this idea about the limits to what marshes could take up, right? We have this idea, marshes are sopping up all the nutrients. But based on what we know was happening, for instance, in terrestrial forests, we know that often ecosystems become saturated in what they can take up. There just becomes a physical or a biological limit to the amount that they can process. So we approached this using a classic ecosystem approach of input-output analysis, and we had to sort of modify it a little bit for a tidal system. So, you know, at low tide, the creeks drain pretty much dry, and then the water comes in and it floods the marsh, and then it goes back out again. So what we needed to do was to measure that incoming flooding water and then the outgoing ebbing water, know the hydrology involved. So this is Ian from Jen Bowen's lab doing some hydrology there. This is Ashley. We had to filter a lot of water, take a lot of water samples, filter a lot of water for nutrients, take more water samples back to the lab, filter them for other things, and then combine that hydrology together with those nutrient concentrations and sit out there for a solid 12 hours. And of course, you can't just do this once. You have to do it a couple times over the course of the summer to develop a net nutrient budget. So how much was retained by the salt marsh? And we also did this using 15N isotopic tracers. So we could say very precisely, this nitrogen, nitrate that we're measuring came from the stuff that we added and is coming back out again, right? And so this is what we learned. This is the reference marsh system here that we use. And there's one of the things that we confirmed was, yes, at low levels of nutrient loading, characteristic of about 5 to 7 micromole nitrogen, marshes took up and retained almost 90% of the nitrogen that fled the marsh. So that is exactly what people thought marshes should do. The problem is that when you looked at nutrient-loaded marshes, where we basically increased the nitrogen loading by a factor of 10, so we're taking it from background to typical nutrient enrichment, the amount of nitrogen retained was only 45% of what we loaded into that system, right? So we've clearly saturated the ability of that marsh to take up the nitrogen. Now, don't get me wrong. This is still a lot of nitrogen, okay? So 90%, I'm not supposed to touch the screen, 90% times 3,000, you know, so 2,700 grams or moles of nitrogen in the reference marsh. 50% of 30,000 is still 15,000. So we increased loading 10 times, but nitrogen retention only increased by a factor of 5, right? So there is a limit. We don't know exactly what it is. We do know that 70 micromolar concentrations that are characteristic of eutrophic coastal systems is too high for marshes to completely deal with. Then the next question we wanted to ask is, well, what happens with all that 15,000 moles of nitrogen that the marshes retained? And so we look at it from 3 different components. One is we wanted to look at landscape change. Because remember, we had this idea that adding nitrogen might increase plant productivity and help marshes keep up with sea level rise. We wanted to look at plant and decomposer communities, and then we wanted to sort of ramp it up through the food web. So this was our basic idea here, right? Was you add in nutrients, the stream-side Spartina increases in productivity, more of that carbon is buried, and so the marsh itself will keep up with sea level rise easier. So that was our going in hypothesis, and as most of you know, we were just flat wrong. That is not what happened. And I think, we should be, I guess, forgiven a little bit for not looking for this because it wasn't exactly what everyone thought was going to happen. We were out there measuring things that would measure the marsh going up. We were not measuring the things that would really measure the marsh going down until about year 5 of the experiment. And we didn't start measuring it until year 5 of the experiment when we actually had the marsh tank was out there adding the fertilizer, and we ourselves kept coming back home at night and saying, I got stuck in the marsh a whole lot more this time, and I thought it was just me getting older, but then the young kids kept getting stuck, and I thought they were just inexperienced until the marsh platform itself, the legs of the marsh platform actually sank into the mud, tilted forward, and slid the entire tank into the creek one day. And that was when we realized, no, we're not making it up. The edges of the marsh were getting softer, they were getting soupier, they were getting more difficult. And we started observing, so this is what the marsh creek looks like, this sort of mud flats, and then this bank. And after about 5 or 6 years, we observed that the marsh had all these crenulations and cracks and chunks that were falling down. So we started measuring those. And again, this is just another view. These are both views of the enriched marsh. And these were just not, these are phenomenological things that we measured, but you didn't see them in reference marshes. And so when we first did the measurements, we had a whole suite of reference marshes that we measured over multiple years. The number of cracks along the side of the edges basically stayed the same. It was in the noise of being able to do the measurement. The first set of experimental marshes that we did sat way up here. So 5% of the creek bank might have had a crack in it. In the fertilized reach, almost 30, sometimes 50% of that creek bank had these huge deep crevices right next to it and marsh chunks that were sort of calving off and falling over. We weren't convinced that we were really understanding this right, so we started a second manipulation. We had to order more trailer trucks of nitrogen from Chile, and restarted the experiment here, taking one of our reference sites and adding nutrients to it, and now being able to track it over time, now that we knew what we should be looking for. And what you see here is that as years of fertilizer increase, the number of cracks and crevices go up over time until it reaches a sort of plateau here, the long-term nutrient ones, which we think is because at this point, all of the Spartina alterniflora has basically cracked off and it's in the Marsh Creek, and we're just left with that edge that's now Peyton's, which doesn't flood with every tide. So the area that was really flooding with every tide is what fell apart. And basically what we were doing is turning that marsh edge into a whole lot of mud. So we did point transects to look how much of the marsh edge was mud, and these are just to show you the variability among the reference sites that we looked at. Less than 5% or 5 to 10% of that marsh edge is normally mud. And what we've done is convert it after 4 years into about 20%, and after 9 years into about 30 to 40% of mud. At that point, we think it stops converting because we've basically lost almost all of the alterniflora along the edge into slumps that are in there. So we're converting it into a much more muddy place. And I think we can be forgiven for not noticing that an already really muddy place got even muddier while we weren't watching. Well, to understand the mechanisms behind this, again, we wanted to look at that balance between plant production and microbial decomposition, because clearly that's what rules what happens in a salt marshes. Jen would tell you it's all about the microbes, and she has a point, at least in this case. So we looked at the plant and the decomposer response. And really what we found for the plant response was only a very modest increase in above ground primary productivity, really across all the years. This is just one example of the kind of data we have. It's called shoot-specific mass, so it's the weight of a leaf per meter squared. The leaves get a little more robust. They get a little greener if we measure chlorophyll. They get a little bit more nitrogen content. They get taller, but it was always relatively modest. And it turns out that we actually are over fertilizing the plants, and so they get a little taller, they get a little heavier, and like anybody who's grown corn and put too much nitrogen on it, it overgrows its structural strength and falls over. This is called lodging. It's something that farmers have to avoid. And so we found that the marsh plants themselves were overgrowing and falling into the creek, so limiting their ability to increase in productivity. The most striking finding that we found was that the plants actually decreased the amount of production they allocated into making roots and rhizomes, so below ground structure. And it's that below ground structure that binds the sediment together, that creates organic matter, that helps the marshes stay up with sea level rise. So, so again, here's a typical depth profile for reference marshes here with a lot of high root biomass very close to the surface. And plants are producing roots because, why do they produce roots? To take up nutrients. Well, you got all the nutrients you need. You don't need to be making roots, right? So they sort of stop their root production. That root production has this other ecosystem function of binding everything together. So they decrease in their root production here relative to the fertilized ones decrease their root production relative to the reference site. And that finding actually was probably one of the most controversial things that came out of our project. I mean, it makes sense to us in retrospect, but at the time the thinking was that Spartina would produce roots and rhizomes in proportional to how much carbon it fixed in the above ground area, right? Because they were always doing that kind of work in systems where Spartina was nutrient limited. So it never had really all that excess nutrient that it could. But after a while, people started looking in other places, and these are just some cores to show you. This is from the Sipawishet Marsh, where Ivan Dalliela has done 30 years of nutrient additions. And indeed, when he went, because he didn't believe us either to begin with, and cored his own experiments, what he found was in the reference, you can see this sort of peaty composition here, roots sticking out all over the place. In his fertilized ones, not as many roots and a whole lot of muddy, slushy, soupy mud, right? So we convinced Ivan, and then we got some help from Kathy Wigan and the EPA, who went out and looked at Jamaica Bay, which is one of the most nutrient eutrophied coastal systems we have in New England. It's got 5 sewage treatment plants dumping all of New York City waste into it. And what she did was to go use CAT scans to look at the density of roots and rhizomes. So these are vertical, looking down at the top of a core here. And in stable environments in Jamaica Bay, where there's less nutrient loading, you can see the roots and rhizomes are really dense. But down here, what you see in the degrading marshes are a lack of roots, and mostly it's in the roots component. The rhizomes don't really change all that much, but it's mostly in the root component. And then we just recently completed a study in Westport, Massachusetts, where we looked at this across a gradient in an estuary itself, across a gradient of nutrient loading from basically near the ocean where the nutrients get diluted by ocean input, so the concentrations are less, up to the headwaters, and found that same kind of pattern of less below ground relative to above ground with nutrient loading. So the kinds of phenomena that we were measuring in the experimental marsh were also being reflected in other systems. So then we moved on. Whoops. Okay, whatever that was I did is not good. There we go. One of the questions that we then asked was, is this just a phenotypic plasticity response, or are we actually selecting for eutrophic genotypes? And again, this work is really work that's just in progress. There's a lot of people looking at this, including Randall Hughes and her group here. But the idea is that with nutrient enrichment, we may be taking that wider gene pool of genotypes and actually converting them to eutrophic types that have these trait characteristics that are sort of maladapted for the ecosystem functions that we expect. And this is the work of Tom Mosder and his colleague, Melissa McCormick at Smithsonian. And when they looked at the genotypes of Spartina, this is the creek bank Spartina here, the place where we saw all the change, in our reference sites and in the nutrient-enriched sites, when they compared the gray dots in both are the pre-enrichment. So we had archived samples, which he pulled out and analyzed. And you can see here that there's some genomic differences using microsatellites between the sites. But in essence, the genomic diversity in the reference site stays the same over the 10 years of the experiment. Whereas in the enriched site, we've shifted the genomic diversity and actually also narrowed we've also decreased the allelic diversity. And so the question is, is this, how fast can we recover from this? Is it associated with those nutrient adapted characteristics of the plants? And both Randall Hughes and Tom Mosder have done common garden experiments, switching things back and forth and looking at both of those characteristics. So stay tuned for the news on that one, but my understanding is this is looking like it's actually a nutrient adapted selectivity for plants with less below ground. So now the decomposers, and this is the part where I get really nervous because this is really Jen's work and all of Jen's students' work. But really the basic fundamental questions that we were asking is, are we stimulating the decomposer community by adding all this nitrate? Are we actually increasing the amount of CO2 production going off into the atmosphere by increasing microbial decomposition? Are we doing this more than we are stimulating plant productivity? And are we creating more N2? So have we stimulated that denitrification cycle, which is related to decomposition, that, here's the equation up here, nitrate plus organic matter produces N2 and CO2. And the work by Tom Mosder and his crew in the last couple of years has shown that we have increased total ecosystem respiration in these marshes by around 30%. So so we've stimulated that decomposer community. We're pumping more CO2 into the atmosphere. We've increased the respiration rate. We don't think this can be due to root respiration because we know we've also decreased the amount of roots, right? So this is a combination. The way they measured this is with the chamber on the marsh, which is a combination of microbial and root respiration. But if your total roots are going down and respiration's going up, it's got to be from microbial. And so Tom just recently completed putting together a model in which he compares that decomposition stimulated CO2 input to the gross production of the plants. And what he's found, and this is very preliminary, is that we seem to have converted marshes from being sinks for carbon, this concept of blue carbon, where they're taking CO2 out of the atmosphere, converting it into plants, and converting it to peat, to actually being a slight source to the atmosphere. So we've completely switched the carbon dynamics from storing carbon to releasing more carbon. Now this needs a lot more further work. This is really something that's just been completed. But it's very interesting if we have actually flipped these marshes from being sinks to sources. It's very consistent with the accelerated rates of decomposition that we've measured from litter bags, from tea bags, from Jen's work on microbial production, but it was that balance against the stimulated productivity. But like I said, the stimulated productivity of plants was only about 15%, whereas respiration was much higher. So Jen has been working on, what is Jen and her students, and this is really a lot of Pat Kern's work, have been looking at, what are the implications for the microbial community. And one of the most astounding things that they've found is that the nutrient enrichment has induced dormancy in these marshes in the microbes, so that microbes here in the reference site, about 50% of the community is 50% active, 50% dormant or inactive, sort of in hibernation, if you will, waiting for the right environmental conditions for them after the first 2 or 3 years of nutrient enrichment. So this is the years of nutrient enrichment across the bottom. These are the calendar years here. After about 2 or 3 years, well, we've bumped that up to about 60, 65%. Yeah, that's interesting, but maybe it's not hugely different. But yet by year 10 and 11, almost 90% of the microbes, the bacteria in the marsh, are now dormant. and we've pushed that community to where 4 of the operational taxonomic units, all of whom do some form of denitrification, were the dominant units that were active in the active community. So we've really changed we haven't changed the total number of players in the system. We've just changed the ones that are actually doing things in the microbial part. This contrasted dramatically with the fungal decomposer response, in which Pat showed that we've not only changed the total diversity of the fungi, but we've also pushed them again towards those communities that are more characteristic of denitrifying communities. So this is across the marsh platform here. This is the creek and low marsh that's flooded every tide, where the blue again is the reference, and so we've increased these are the percent of the community that were denitrifying fungi. I don't think we even actually understood that marsh fungi did denitrification before Pat did this. So that was astonishing to a lot of people. But it goes from, virtually none up to about 10% in the mudflat. In tall spartine alterniflorida, again, 5 up here to 20%. Still a change in the high marsh, but interestingly, it's sort of proportional to the flooding, right? So the creek and the creek bank marsh is getting flooded more often, the high marsh not so much. So we don't see quite as much of a change there. Well, interestingly, all of this combined together, this role of it as a denitrifier, the really high levels of denitrification that we know is occurring in tidal mudflats, but really particularly in creek marsh edges. This is some work by Ann Giblin, who's measuring the actual rates of denitrification here, where she's shown that the creek edge marsh here is, 2 or 3 times more active in denitrification. So if we're losing that, we're pushing the community towards more denitrifiers, but we're taking the ground out from underneath them. And so the net result is we're probably losing that ecosystem service of stripping the nitrogen out, because the sites where the most active denitrification is occurring is in that creek bank that we're losing really rapidly. And again, how that per area versus changing the microbial community, all that plays out is something that we have to think about in the future. So what about the food webs, right? What about all the animals that are living in the site, and what are they doing, and how is this affecting them? So this is my mummy chog here. Cute little fish. This is actually probably the largest mummy chog anybody has ever seen. They're usually about half that size in a salt marsh. This is one of our REU students that worked on Plum Island a couple of years ago. And what we did was to look at the food web that sort of leads up to the mummy chog. So we looked at bacteria, algae, the small meiofauna and the macrofauna. And this is some work done by John Flieger from LSU, Pierre Pascal, who was a postdoc who came over from France, and David Johnson and Kerry Galvin. A whole host of people worked on this component of the system. And we also, one of the things that we showed was, yes, not surprisingly, we stimulated the algal abundance. Turns out we didn't stimulate very much, which kind of surprised us. We thought we might turn it into like glowing green creek marks, but we didn't. This is an assessment of diatoms in the marsh here based on algal cell counts on glass slides that we put out as a way of monitoring it. So clearly we increased the algal productivity here, particularly in the later years, right? So across the board, we're sort of seeing this cumulative impact that moves up the food chain and impacts things where after a few years, we saw some differences, some of which went in the right direction, some didn't look too different. But then in years 8, 9, and 10, we start seeing the cumulative impact of nutrients in both the microbes and their dormancy, but also in the algal response. So then we looked at the inverts across all of these marsh habitats. And I'm not going to show you all of the habitats and all of the species. This is just an illustration of work done by David Johnson here, looking at the marsh surface, and again, comparing reference to fertilized for 3 of the key species that operate as fish food for mummy chogs. And what we see there is we've increased their biomass on the marsh by about 30%. Again, 30, maybe in some cases 40%. So we've increased not only the algal production, but we've increased the inverts And we've also increased the microbial production. So one of the things that we wanted to look at was, did we differentially affect whether it was the microbes that are chewing on the decomposing marsh plants versus the algal production that was supporting the food web. And so Pierre did a double tracer addition experiment where he labeled the microbes that were decomposing the Spartina versus the algae using 15N and 13C, and then sort of tracked it through the food web by combining grazing rates with biomass to look at how this system had changed. And again, as you can see, through time, if you look at this graph down here, which is sort of a summary of all of this right here, this actually did this. Each of these boxes is like 5 or 6 different little critters, and he put together a little network model of looking at everything. But what you see here is over time, we see an increase in the amount of both algal and microbial organic matter sources that are moving through the food chain. So again, that cumulative impact here. But surprisingly, what we didn't see was any large changes in the proportion. In the reference site, there's slightly more actually algae that get into the food web from the reference site, which is a little bit backwards from what we thought 15 years ago. We thought these food webs should have been run by marsh detritus, and certainly they use marsh detritus, but algae is really important. We stimulate the algal production. We get about a 50-50 split after about 1 year, maybe a titch more algae in year 6, but still a pretty good contribution of that bacterial production into the food web. So we're stimulating the microbes, we're stimulating the algae. That's moving up into the inverts. Is that moving up into the fish? And so we sampled fish in this system using a variety of techniques. We started out getting really muddy and doing seining again, and gave that up after about a year because we were getting too stuck. And developed this technique where we use flume nets that measure the fish that move from the creek up into the marsh, and we can get these estimates of fish per meter squared, something I was dying for when I was doing my undergraduate thesis, but didn't have. Now I do. And what we find is that, again, we see this bottom-up push. This is the ratio here of the biomass of mummichogs in the enriched zone compared to the reference zone. So anything above this line right here says we have more mummichogs in the enriched sites than we do in the reference sites. And again, we started out actually with slightly less production in the randomly chosen site that we enriched. But over time, that ratio increased until in about the 6th year of this experiment, we had just 3 times more mummichogs in the enriched zone. So we figured we're good to go here. We've got a classic bottom-up push with stimulating the phytoplankton and the benthic algae and the microbes, and it's moving through the inverts and it's making up to mummichogs. And so then I made the biggest mistake of my career. We kept going. And actually, for those of you who know him, I hired Jimmy Nelson from Florida, who studied gag grouper, this huge fish. I figured if anybody could figure out about the fish and sample them well. I teased him the first couple of years. This is when I hired him. This was his first year at work. So there was a cumulative impact of Jimmy on the marsh. So really, the big question was, what the heck happened, right? Because what the heck happened? And we had 2 competing ideas. One is that whole food web could collapse because maybe we'd driven it to anoxia, although the DO-meter said we hadn't. But, we could check. Or one of the things that we know is really critical about salt marshes is that mummichogs sit at the top of a food web that couples the salt marsh creek area together with the marsh platform, right? They move from the creeks up into the platform, they feed, and they come back down. And we were wondering about all that cracking along the edge, and maybe that, maybe we've actually spatially decoupled these food webs for some reason. So we did some calculations to take a look at that. And what Jimmy did was to take all of that invertebrate production and run it through a trophic network model, food web model, and basically turn it all into mummichog equivalents, right? And so again, if we look here at the ratio of the reference well, this is the reference, whoops, and this is the fertilized. And if we compare how much fish production we could get if mummy chogs ate 100% of the inverts that were there, how much fish production could we get? Anytime the number is below this zero line means we're in a deficit. We don't have enough invertebrates to support all the fish that were actually in the system. And so, as you can see here, in some years we're well below that deficit. In other years we sort of bump around the zero line. But remember, we've said that mummy chogs can eat 100% of what's out there. And we know that's not true because there were other fish out there and shrimps and crabs eating on those things. It's only when we add those high marsh epiphanas into this equation that we get enough productivity at the invertebrate level to support the kind of fish production that we have. Far more invertebrates are available, suggesting that mummy chogs are perhaps not food limited as long as they can get there. But what it also says is that high marsh area that's only flooded 5 to 10% of the time is really critical to these food webs. But again, if we look at the productivity of the inverts relative to the fish production in this collapse period in the fertilized creek, we still got lots and lots of invertebrates. So that's not the issue. We haven't collapsed the food web. We haven't run out of food. And so that left us asking the question, is it something about that connection between the creek and the marsh that we know is all crenulated and falling apart? And so we took a look at this by doing, this is the reason that you have REUs work for you, because they give you really cool graphics. This is what we know mummy chogs do and the reference conditions. And our question was, what happens in this crenulated condition here where these lumps and bumps provide interesting locations for predators to hide and attack and feed on mummy chogs? Is this all this lumpy bumpiness a physical barrier, or is it predator avoidance? Is it changing their behavior at all? And so we had an undergraduate student do a thesis where she looked at the behaviors of mummy chog and the energetic demands of mummy chogs, comparing this kind of creek edge to this kind of creek edge in an experimental system that we created in 5-gallon aquariums, sort of mimicking the two sides. And what we found was that most of the behaviors of mummy chog that are very classic, their bottom hunging behavior, their schooling behavior, didn't differ between the reference and the treatment, or the reference and the fertilized. And the only thing where we saw a huge difference was really in the kinds of behaviors you associate with predator avoidance, right? So they're hiding in the grass, they're pausing before they're going over edges. She marked all of these things down. And when we did an energy budget for how long it took the fish to get to food we'd placed on the other side of the creek, it took them much longer because they're going up and down and they're stopping and they're looking to get to the other side of the creek, so that the energy demand of operating and being on the hunt or being hunted made their energy demand almost twice as high in the fertilized section. So we think that it's actually, we don't have this one completely worked out, but we really think it's more or less that decoupling of the creek from the high marsh via this change in the physical structure of the landscape mediated through the behaviors and responses of the fish that needs to couple that landscape together. So we have this decoupled landscape here that's resulting in less mummy chog production. Well, why does anybody care if there's less mummy chog production? Well, because we all like striped bass. And so Jimmy did a very interesting calculation, and he showed that, in the reference condition, under good conditions, the marsh produces about 4.5 grams of mummy chogs per meter squared per year. We got about 40 kilometers squared of vegetated marsh in Plum Island. So that scales up to be about 4,475 legal striped bass that this marsh produces every year. If you cut the mummy chog production by a factor of 5, which is basically what we did, you're going to cut that food web link between the marsh and the larger open bay system and lose the ecosystem service of supporting all those fisheries, marsh-dependent fisheries as well. So it's not just that you care about mummy chogs, you care about these bigger fish too. So that's kind of the highlights of the tide experiment. There was a lot there. the take-home message is that really, you really have to think hard about whether or not you can add up all the pieces of an ecosystem and put them together and really understand what's going on. You really have to do large-scale experiments at the scale relevant to the organisms that are involved. This was the first experiment to really show that eutrophication could lead to marsh loss. It was the first experiment that also showed that there were limits to the landscape ability of marshes to take nutrients out of the water, and that most of those areas that we declare as eutrophic, because they have fish kills and those kinds of things, are exceeding the limits of marshes. We don't know exactly what the shape of that curve is, but we know that 70 micromolar is too high. We also know that it induced a lot of changes in two of the key components. The microbial diversity, we pushed it to, didn't change the total diversity necessarily, but we pushed it to a system where only a few of the microbes were operating. Plant diversity, we seem to have done some selection there, pushing them again towards ecotypes, if you will, that were maladaptive at the ecosystem level in providing those ecosystem functions. And we changed the big picture ecosystem production functions of nitrogen retention and secondary production. And one of our concerns is that because of these kinds of changes in here, we may well have changed marshes to the point where they're going to have a hard time to recover. And so for the first time in 13 years, we are not going to toast adding nitrogen to the marsh with mimosas in May. We have decided that really as scientists, it's our responsibility to provide the information that we need to understand how ecosystems can recover from the stresses that we've induced. Could we learn more by continuing to fertilize? I'm sure we could. But I think we've got the big picture down. We know it's not good. And what we need to do now is provide the information across that entire ecosystem for how long it'll take for it to recover and what it will take. One of my concerns is that, there were 7 billion people in the world when we started the TIDE experiment. So 4 when I was at Northeastern, 7 when we started the TIDE experiment, and pretty soon there will be 8 billion people in the world in the year 2025. So really, what do we need to do to achieve a world where nature and people can both thrive? Because it's clear we need nature to thrive for humans to thrive. You're not going to have fish to fish if you trash the ecosystems they live in. Those ecosystems won't be able to provide those odd ecosystem services we never thought about before, like protecting coastal cities from storm barriers, unless we can figure this one out. And there are plans afoot to restore coastal salt marshes, for instance, in Louisiana, that will take millions to billions of dollars to bring back that blue carbon, to bring back that fisheries production. It's a daunting thought. But I personally believe that as a society, we're up to the challenge. And I take heart in Boston Harbor, right? 1982, dirtiest harbor in the United States. Harbor of shame. Influenced the presidential election, for God's sakes. Nothing does that anymore. But fast forward to 2015, and Boston Harbor beaches were declared that Boston Harbor beaches are cleaner than the beaches in Waikiki. Cleaner than the beaches in Waikiki. And for all those undergraduates out there, I say, that's a testable hypothesis that I'm going to go test. But this should also give you hope for the fact that we can do it, that we are up to the challenge. And I say, as it took science to figure out the problems in Boston Harbor, no one wanted to believe it was us. Nutrient loading was supposed to be an agricultural issue, not a city issue. But now we know it's a city issue. It took science and engineering to figure out the path forward, right? And then we executed it. There's as green building architect Bill McDonough has said, and is famously quoted for, you know, the Stone Age did not end because we ran out of rocks. The Stone Age ended because people wanted to live a different life, right? And I think that that's where we are right now in society. We need more science, not less science. And I think, you know, I'm also very proud of the changes that I've seen in talking to all the people here at Nahant. You know, when I came here as an undergraduate, not dissing you, Dr. Reiser, but worms were it. We did a lot of work on the ecology of little organisms. And as I went around talking to all the students today, you know, I heard about sociology, I heard about fisheries issues, I heard about microbes and genomics and ecoevolution and adaptation. I mean, the breadth of things that are being done here at Nahant are just amazing. And the new Coastal Urban Initiative, sustainability initiative that Northeastern is undertaking, where they're going to bring social scientists together with biologists, together with engineers, to try and craft a new way of living in the coast, I think is really a far forward-looking approach. And I would also say to all the students out there, you know, at a time when our science is under a microscope and in a spotlight, this is when we need more science, not less. And I implore all of you to get away from your microscope, put yourself in the spotlight, and tell the story of how your science is helping us change the world. Thank you. And if you have any questions about the genomics, ask her. I would love to have somebody come do that. So, you know, we have thought about those things. And like I said, we've had 100 different scientists, grad students, undergraduates do lots of different things. It is a platform. Anyone here who wants to come help us understand how those things might change is welcome. I think that's an excellent idea. So, yeah. Yep.
We've got a very serious drought condition now over the last half
Yes.
year, and the flow of the Rouse River started some very problematic.
Yep.
So, is the salinity of the marsh goes back to the state?
Nope. So, I mean, we have YSI monitoring equipment out there, and it's very interesting that because this system is really pretty marine dominated anyway, it's really, the salinities bounce between a low of 20 and a high, the flooding water, a low of 20 and a high of 35. Mostly it's between 25 and 35. And that really didn't change with these recent drought conditions. Now, what may have changed a little bit is the soil salinity. Online audience. No. Yeah. So, you know,
So from up